Okay, what points of this do we want to describe when we're explaining what transcription is? What are things you need to highlight? In general, let's do a general thing first. What would you say? Okay, that's it. DNA. That's the answer. All right. Next question. So in general, all I'm doing is copying a gene off the DNA to make one particular protein. So first thing you need to do is unzip it. Then add. So when we add a base, an RNA base to copy this, what do we call that base? It's got a name. It's a something 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 base, one word. Yeah, so we're going to throw on the complementary RNA base. And it we'll just go, let's say closes DNA as it copies and zips at stop codon. All right, so we unzip it, we add complementary bases, we close it as we're copying, and then we zip it completely up when we're finished with the stop codon. Then we need to do something to it before it can leave the cell, which is what? Yeah, we need to remove introns before leaving nucleus. Because right now it's actually, with the introns in it, it's nuclear RNA. And we wanted to leave as extrons only. And then that'll make it messenger RNA. All right. Translation. Why is it called translation? The transcription one was kind of obvious. Why translation?
Translate the language.
Okay, so nucleotide language. Translating that into amino acid language. All right, so we've got messenger RNA. It's going to attach to half the ribosomal RNA. Then first transfer RNA not arrives. Let's go with attaches to the first messenger RNA codon. And other half of ribosomal RNA caps the transfer RNA, and now I've got a full ribosome. Then second transfer RNA attaches to messenger RNA. Then first transfer RNA hands off amino acid chain. I guess in this case it's only one, but it's the beginning of the chain. Hands off the amino acid chain to second transfer RNA. First transfer RNA leaves. Third transfer RNA arrives. And then we repeat. Then we go back to that. And this continues until you reach the stop codon. Then everything disassembles. When you're finished with this, you have a primary sequence of amino acids. So you have your primary structure. And because the primary structure happened in the cytoplasm and happened there and coming out of a ribosome, it starts to secondary and then tertiary fold once it's completely finished. And if we could get that same kind of thing to happen when we put those things together, it would be great, but we haven't been able to figure out how to do that. PCR. I have a container with DNA. Nucleotides and enzymes. I heat to I separate out the DNA strands by heating it up. Then I cool to have the enzyme. Attached to the DNA, and then I heat to speed attaching the complementary bases. Then I repeat. Yeah. Why are you biting your lip?
What exactly do you mean by the primer sequence?
Primer sequence is the part that allows the... You can't just open up wherever the heck you want, and so the primer sequence allows the copying to begin. So we don't have a start codon with proteins, we just have a stop codon, but for DNA, we need a place to start and we need a place to stop.
So that's the primase?
Yeah. So I have two enzymes in there, one's primase and one's the DNA polymerase.
And what's the TAQ polymerase?
That's a better name than DNA polymerase.
Oh, okay, that is basically.
It is DNA polymerase, it's just a more complete name. And what about the protein synthesis? Didn't really matter which one you picked, but, oh no, I said laboratory one. Okay, so that lab one, I've got, I've got 20 containers of protected amino acids. Deprotect solution and water. We're just inserting this insoluble solid to the first amino acid container, and the C terminus attaches. Then I deprotect. I think I could spell protect. Wash, and then move to second amino acid, and then it attaches to the first amino acid. Then I deprotect, wash, move to third amino acid. So
Like the first and second, third join and water as a byproduct.
yeah, you'll have water as a byproduct because you had carboxylic acid on one side and amine on the other side for sure. Because each time we're doing this, we're making a peptide bond. It's a primary sequence of amino acids. And then we just continue this until all the amino acids are sequenced. And then we remove it from the insoluble solid. Why don't we use this anymore?
Slow.
It's just too slow. That's all there is to it. It's just too slow.
I don't think it's the full.